Hi, my name is Anne-Marie Parker. I'm a research associate on the Joplin campus of KCU. I'm going to be introducing Dr. Angela Pierce for this Faculty Friday Research Seminar talk. Dr. Angela Pierce completed her postdoctoral and doctoral training at the University of Kansas Medical Center in Kansas City campus, where she earned her Ph.D. in Neuroscience from the Department of Anatomy and Cell Biology. In addition, Dr. Pierce is also a board-certified chiropractic physician and a graduate of Palmer College of Chiropractic in Davenport, Iowa. She completed her undergraduate studies at Missouri Southern State University with dual degrees in biochemistry and Spanish. Dr. Pierce has earned multiple honors and research presentation awards. She is a Madison and Lila Self Fellow alumna and currently serves as a governor for the Society of Self Fellows, the Fellowship's International Alumni Association. She has experience teaching molecular and stress neurobiology, neurophysiology, and endocrine and renal physiology. Currently, Dr. Pierce is an assistant professor of physiology and director of COM Preclinical Assessment at KCU-COM. Her current research interests are in chronic pain, anxiety, depression, and exercise, which she will discuss today. In her spare time, she enjoys racing sports and intends to complete a Half-Ironman in 2023. With that, the floor is yours, Dr. Pierce.
Thank you very much. So this is going to be our visual aid going through the talk of what we're going to talk about. To get into the research, I need to begin with a discussion of stress because that is the overall theme of this project, of all of my research really. So there are three categories of stress. We term stress as either positive, which a positive type of stress is characterized by brief increases in heart rate, short-term elevations in stress hormones. Tolerable stress, which is more serious, but a fairly temporary stress response. And the effects of that stress is generally buffered by having supportive relationships in an individual's environment. And then we have what's called toxic stress. Toxic stress is characterized by a chronic activation of a stress response, and that is ongoing in the absence of supporting relationships. So a few examples when it comes to, say, stress experienced by children. Examples of positive stress might be getting overly frustrated, going through the process of having to get an immunization, a first day at daycare, first day of school. There's an opportunity for a child to learn adaptation, especially in the environment of having a good supportive relationship, particularly at home. Tolerable stress examples could be either a greater magnitude of threat or adversity. It could be something serious like the death of a family member, undergoing serious illness or experiencing natural disaster. But what makes tolerable stress, it categorized as such, is again, this buffering of the stress response by this supportive caring relationship. Examples of toxic stress I'll talk about in a minute. This is important, particularly in the context of children, because children's brains are growing at an exceptional pace. And in fact, you may not know this, but by the second year of age, 80% of our brain growth is essentially done. And that's shown here on this chart. You see the age of an individual in years. And there's this explosion of growth that takes place in the first few years of life. And in fact, as characterized by this figure on the bottom, it has been shown that there's 1 million new synapses every second that takes place in these first few years of life. And this graphic is really pretty and nice because it shows the just explosion of what we call the dendritic tree and the complexity of that those synaptic connections. And then you can see that by 2 years, there really is a peak in the connectivity within the brain. And then through natural pruning by the adult, these synaptic connections have really stabilized. So the purpose of the reason why the type of stress that we experience early in life that's important because it has an impact on this constantly growing development of the brain. But when it comes to stress, you have to train the brain. It's not just born with this kind of ability to handle stress, process it, and then move on from it. Beginning in utero, the brain gets little exposures to stress over time. And so I kind of equate that to training to run a marathon. You wouldn't just step out your door and probably be able to run a marathon. Everybody has to train for it. You have to build up for it. build up to it. And so the same sort of thing happens with the brain and training the brain to handle a stress response. And that is called the critical period of neurodevelopment, okay? That critical period of neurodevelopment is characterized by a buffering of the stress response. So if you think about an analogy of being around a loud noise that might otherwise cause some damage to your hearing structures, if you wear earphones, that can be protective to otherwise noxious stimuli. And so during the stress hyporesponsive period, which is that critical period of neurodevelopment where the experiences of the brain have a lifelong impact, there is a natural inborn protective mechanism where the stress response is buffered. And that buffering is important because without that buffering, that differentiates the toxic stresses from tolerable stresses from the positive stresses, okay? And so if you have toxic stress, it has been shown that that toxic stress experienced during the critical period of neurodevelopment can have a lifelong impact. And that sets the stages for findings that not only persist throughout the life of that individual, but recently has been shown to have a generational response as well. It can have an impact on overall general health, regulation of many, many gene expression patterns, and even impact morbidity and mortality. So we come back to that definition of toxic stress. What are toxic stressors beyond stress experience without that nurturing, buffering response? Well, what comes to mind is probably major things like these on this screen. And certainly these are very dramatic examples of toxic stress, you know, experiencing war, natural disaster, famines, experiencing extreme accidents. All of those things are certainly very dramatic levels of stress. And, but more disturbingly, early life stress can have much higher levels of being more common than you might even realize. And so whenever we experience these kind of toxic stressors or their equivalents early in life, we call that early life adverse events. And while these things are certainly terrible and can have a detrimental impact on any growing brain, it's not only these dramatic examples. It could be things that are much more common, such as neglect, exposure to abuse, or even premature birth. How does that happen? What are those signals? And the main signals involved are shown here. There are 4 things that are primarily involved in this, the molecular components of the HPA axis. The first is corticotropin releasing factor itself. And then there are 3 related neuropeptides, and they are termed urocortins. There's urocortin 1, urocortin 2, and urocortin 3. CRF and urocortin 1 signal through the CRF type 1 receptor. They will bind both to this receptor. Urocortin 2 and 3 will also bind the CRF type 2 receptor. Urocortin 1 actually has affinities for both. Now, there are 2 different receptors because there are 2 different effects that are mediated through this signaling pathway. The CRF type 1 receptor signaling pathway results in anxiogenic behaviors. These are anxiety-inducing behaviors. It essentially ramps up anxiety, mood, and the HPA axis. It perpetuates the HPA axis through a positive feedback mechanism. The CRF2 signaling pathways are essentially the opposite. They are anxiolytic. They reduce anxiety. They reduce activity of the HPA axis, and they do so through mediating negative feedback on the HPA axis, and that involves the hippocampus. Furthermore, the CRF2 pathway has been shown to inhibit the CRF1 pathway. So another way by which there is a negative feedback on the HPA axis. And then finally, we have the receptors for the glucocorticoids, the mineralocorticoid and the glucocorticoid receptor. The MR and the GR have very important effects body-wide, but in the context of the HPA axis, they can actually inhibit corticotropin releasing factor back up at the level of the hypothalamus. The MR and the GR are important for mediating the cessation of HPA axis activity upon the conclusion of a stressful event. So going back to my roadmap here, I'm going to talk first, now that we've kind of set the stage for stress, I'm going to talk first about some data on how stress, particularly early life stress and adverse childhood experiences, can impact pain, specifically chronic pain. So that's what I'm talking about first. Question then is, how do we study this? How do we study the relationship between adverse childhood experiences and, say, a chronic pain syndrome, or in what I specifically studied was chronic pelvic pain syndrome? Well, we do so using animal models, and that's what I did. I used a mouse model of early life stress, which is widely accepted in the field, known as neonatal maternal separation, where essentially after mama mouse has a litter of pups, we remove the pups from the presence of the mama mouse for 3 hours a day throughout their entire postnatal life. So essentially from the day after they're born and until the day that they would be weaned, which is equivalent to adolescence. And then we study the adult pups. So after the pups grow up, we can study the adult pup behavior. Of course, this is no longer a pup, this is a mouse. And one of the ways that we study pain in mice is through behavioral responses to different stimuli. This is an example of a mouse that has been anesthetized and is undergoing a surgical procedure. It's a relatively minor surgical procedure whereby we surgically implant electrodes into the abdominal muscles via a small incision in the abdominal skin. Those are surgically implanted, and then the electrodes are kind of routed through the skin and then externalized at the nape of the neck. And then we can induce various stimuli and then quantify using the electromyographic responses a reflexive abdominal muscle contraction, essentially, that has been shown and is widely, again, believed to be reflective of a behavioral response to pain. So essentially, in this example over here, we have a mouse that there's been a stimulus applied, and then the EMG trace activity is shown on these traces here. And you can see that as the pressure of our stimulus increases, there is a greater EMG response, indicating that there is a greater response to pain. And we can extrapolate painful sensitivities out of this data. And so using this surgical model, we specifically, I say we, but what I did was I took mice that were from this neonatal maternal separation cohort of mice, and then I take the kind of a normal control litter, let them grow up into adulthood, and I specifically studied females here, and I'll talk about female versus male later. And then as adults, we can use a balloon distension paradigm to measure organ sensitivity. So essentially what we do is we insert either while they're awake or lightly anesthetize a balloon into different hollow pelvic organs, and then deflate or inflate the balloon to various pressures that are measured in millimeters of mercury, shown down here on the x-axis of all of these graphs. The graph on the left is indicating bladder sensitivity. So we have a, on the y-axis, percent of baseline EMG response. So this is the muscular response or the contractile response during the balloon expansion. And we measure that. And we compare our control mice in the black lines to the mice that underwent neonatal maternal separation. Again, that happened as pups, and we're studying the adults now. You can see that the bladder tissue and the vaginal tissue of these mice were acutely hypersensitive compared to their control counterparts, as indicated almost all of the pressures that were studied. So they have a greater response, greater muscular contraction, if you will, during these stimuli compared to their naive counterparts. Interestingly, in the female mice, there's a differential pattern with the colon in balloon distension. In fact, they were hyposensitive in the colon from our female mice compared to their naive counterparts. So we found this interesting pattern of pelvic organ sensitivity was organ specific, depending on the sex of the mouse. I also studied somatic sensitivity, so essentially paw sensitivity, face sensitivity. They're overall very similar findings. I'm not going to show those today, but they're just sensitive. Adult stress also exacerbates the findings that were experienced in early life. So if you have our early life stress or the neonatal maternal separation model, let them grow up, and then you can test their sensitivity to various types of stimuli. They're experiencing quote unquote more pain than their controls. But if you induce a second hit, another major stressor, as adults, what is the consequences of that afterward? And this is something that happens and is studied in humans as well. It's well recognized that experiencing a major life event in adulthood can trigger or uncover an underlying syndrome that may have always been there, but has now been kind of triggered or released, if you will. And so we kind of model that using an adult stress in our mice. And the stress that we use is called water avoidance stress. There's a little picture down here of, essentially, it's very simple. You have a pool and there's a little platform in the middle of the pool, and you put the mouse on the platform. And if they jump off the platform, they get wet and they don't like it. They'll swim around and then they'll get back on their platform. And they stay in there for a certain amount of time. And that is inducing an adult level of stress in the mice. So if I repeated, say, the bladder sensitivity study, and I found that our mice that underwent early life stress were, again, as shown from the data here on the left figure, exquisitely more sensitive compared to their naive counterparts when it comes to bladder distension. You put them through the adult stress and then study them the next day, and then wait a week, and you come back and you study them again. And a week later, they are even there's even a greater robust response to the bladder stimulation compared to baseline and compared to their naive counterparts. So the adult stress differentially impacted the mice that had the early life stress compared to control. And one of the findings, if you start looking in the organ tissues out in the periphery, and this is going to be representative of in the female mice bladder and vaginal tissue, we found that mast cells were greatly impacted by not only early life or neonatal maternal separation stress, but also the adult stress. So mice that underwent the early life stress had, 60 to 70% of the mast cells that were counted in their bladder tissue were degranulated, compared to 20 to 30% in our control mice. Now, adult stress increased the percent of those mast cells that were degranulated a little bit, but it was not significantly different from baseline. But the neonatal maternal separation stress, even after the adult stress, those findings persisted. So you can see a nice, beautiful picture here of a toluidine blue stained bladder. The naive or the control mice are on the left, and you can see all of these wonderful dark purple dots are mast cells kind of sitting in a blue stained bladder tissue. And you have this nice, intact, well-defined border mast cell. And you can compare that to the bladders from the mice that underwent our early life stress. Again, these are adult mice. Their mast cells were, degranulated to nothing, basically. Sometimes the degranulation states of these mast cells were so exploded, it was sometimes hard to find where those granules originated. If you look in the brains of these mice that underwent neonatal maternal separation, either by itself or in combination with an adult stressor, you can find some unusual patterns of expression related to the glucocorticoid and the mineralocorticoid receptor. This is data specific to the hippocampus, but I have also looked at data from many different parts of the brain, and I'm really kind of highlighting our highest yield data here. But if you recall, the glucocorticoid receptor regulates that negative feedback of stress upon the HPA axis. And that purpose is to stop the stress response at the cessation of that stressful experience. But here you see that early life stress, indicated in this white bar, kind of chronically downregulated glucocorticoid mRNA expression, suggesting a disruption in that negative feedback regulation. And adult stress tried to increase that mRNA expression, but it still remained a significant overall effect of neonatal maternal separation compared to their control animals. Contrast that with the mineralocorticoid receptor. The mineralocorticoid receptor is kind of an interesting receptor. It's thought to be more involved in the regulation of the tone of the HPA axis kind of on a daily basis. So the normal diurnal or the circadian rhythm is thought to be regulated by MR signaling. And so you can see here that both early life and adult stress downregulates mineralocorticoid expression compared to control, suggesting, again, there is a chronic exposure to stress. Here's a bigger picture of that water avoidance or the adult stress in mice. And you can just look at this little animal and you can tell based off of the fur, the ear, the tail, that this is animals not really liking the pool. Now you might be asking, what about the cortisol response? What about the physiologic stress endpoint? Well, I also measured that. I've done that many, many times. And there are some kind of really interesting things to keep in mind when it comes to studying blood or serum levels of cortisol, or in mice, we don't study cortisol, we study corticosterone. But if you think about increased negative feedback on the HPA axis, it should result in a rapid blunting or rapid decrease in ACTH, and subsequently a rapid decrease in cortisol. But here, we view findings that are consistent with that decrease in mineralocorticoid mRNA expression, particularly in the hippocampus following early life and adult stress. The HPA axis blunting was more pronounced in neonatal maternal separation mice at 1 week. So this is 1 day and 1 week following that adult stress. Okay, so you can see that this was a significant overall interaction effect and a significant effect of the adult stress. And this is, again, consistent with the patterns that we observed with the negative feedback regulation in the hippocampus. So the mRNA expression, not only of MR and GR, but I also looked at corticotropin releasing factor receptor type 2. If you recall, that receptor is responsible for mediating negative feedback. It is anxiolytic. It's generally thought to be kind of quote unquote the calming receptor. And you can see here the mRNA expression patterns from the hippocampus of these mice show that adult stress increased the mRNA significantly in both control and maternally separated mice, but there was a significant effect of maternal separation compared to the control at 1 week post-stress. But even clearly, this was not able to overcome the disruption to the overall negative feedback that we have observed through serum corticosterone findings and the downstream behavioral findings. Okay, so that was a lot. I'm going to take a moment and shift now and talk about the male mice, because everything to now has been female mice. And there is an interesting divergent pattern with males. First, when we look at maternally separated mice in red down here on the bottom of this graph, this is a findings representative of prostatic sensitivity. Okay, so it's technically the perigenital sensitivity because we're testing the perigenital area. And in mice, it is thought that that is reflective of prostatic sensitivity, but I don't have access to test prostatic sensitivity. It's not a hollow organ. I can't necessarily test it with the same type of stimuli you would test other hollow organs. So we test perigenital sensitivity instead. And what you see here is a graph of withdrawal threshold. So essentially, how much stimulus does it take for the mouse to respond? And so if you apply a stimulus to the maternally separated mice, they respond with just barely a touch, and indicating that they're exquisitely sensitive. Whereas the control mice or the naive mice, either at baseline or even after adult stress, it didn't really bother them. You could apply all the stimulus that you want and they didn't necessarily respond. So our maternally separated mice that underwent that early life stress, very sensitive in the male perigenital area. If you look at the mast cell degranulation patterns in these animals, you see similar findings to what I showed before. And these are going to be specific to the bladder and the prostate with mast cell degranulations that mimic the mast cell degranulation patterns observed in the females. I'm going to summarize our kind of early life stress impact on pain by just saying that early life stress definitely results in findings consistent with visceral and somatic hypersensitivities. And I've only touched the surface of some of these findings. It disrupts negative feedback or evidence of disruption of negative feedback on the HPA axis. We also, I only touched on the mast cell degranulation process, but I've also looked at a number of other neuroimmune signaling mediators within the viscera, whether it's inflammatory mediators, cytokines, expression, mast cell growth factors, even mast cell differentiation factors. There is consistent evidence that all of these are affected by early life stress within the findings and kind of the pattern of our pelvic organ sensitivity, which speaks to this following bullet point, that there's a differential impact on the male versus female mice. And I didn't even get into the colorectal sensitivity in the male mice as well. So next I'm going to talk about the impact of exercise or providing an intervention to this model and what happens. So the next kind of phase of my studies began to investigate exercise, and can the introduction of running wheels either in the at the age of 8 weeks of mouse life, which is essentially kind of late adolescence, or in the beginning of adulthood, or 12 weeks of adult life, what impact does having access to a running wheel change any of the previous findings that I've shown in terms of pelvic organ sensitivity, mast cell degranulation, brain changes, etc.? Well, what I found, and I only have a slide or two about this, but this, you can imagine this is years of work, is that exercise significantly improved pelvic organ hypersensitivity, and it does so in the females and in the males, and it does so regardless of the organ that you test. These are findings consistent with urinary bladder sensitivity using bladder distension protocols, but it can not only kind of reverse those findings. So if I take neonatal maternal separation animals, test them, and then give them running wheels for a month or two, and then come back and retest them, it will quote unquote reverse those findings. It will improve their sensitivity. But if you give them running wheels right after weaning, before they have grown into adulthood, it will prevent it completely. So it's really fascinating work here. And then this is just one graph that shows that animals from bladders that were either remained without running wheels or had access to running wheels, even the mast cell degranulation states improved in these organs. Here is just a very small snippet of the somatic findings. This is forepaw sensitivity. So this is essentially before and after giving them access to running wheels. So this is evidence of if you have maternally separated mice and you test their somatic sensitivity at baseline, give them running wheels for a couple of months, and then come back, you'll find that and retest them, the maternally separated mice that remained sedentary, their hypersensitivities persisted. But according to this, you can look at the kind of the open square here. If you gave them access to the running wheels, it reverted their sensitivity state. Now, something I will note here is this black line with the filled in black circles. These are representative of control mice that remain sedentary. So these are animals that had at baseline, they were fine. You could apply stimulus and to a pretty great degree and they wouldn't respond. But after a couple of months of them remaining sedentary, their sensitivity actually increased a little. And this kind of kicked off a whole other research interest of what is the impact of sedentary caging in these animals compared to having access to a running wheel. These are findings from the Dura mater. So as I said, I've looked at a lot of different parts of the mouse, and part I was really getting interested in was findings indicative and consistent with the migraine experience in humans, of which mast cells are thought to play a big part of the migraine pathophysiology. And so I started looking at maternally separated mice and their mast cell degranulation status within the dura. And again, these findings are consistent with other parts of the sensitive organs, if you will, particularly those pelvic organs that were reverted by exercise. Okay, the last part I'm going to talk about is reward. So I've also found that access to running wheels and the impact of early life stress does either disrupt or improve behaviors that are thought to reflect the reward system kind of milieu, if you will. So what I did was I gave mice caging conditions where they have access to just regular drinking water and then a kind of a sugary sweet water. And so the sucrose water, if you will, is very stimulating of the reward pathway in a mouse. And a mouse that is not really interested in drinking the sucrose water is thought to have a disrupted reward system. And so what I found was the preference of our animals in drinking our sucrose water differed according to exercise and early life stress status. Although it was actually, the findings were funny, it was actually backwards from what I thought was going to happen. And in fact, the early life stress animals preferred to drink the sucrose water more than their control counterparts. So they're just chugging the sugar water all day long. Whereas the control animals, I mean, they would kind of go back and forth more. And so I started to think about this, and I was thinking about, the last time I was really stressed, what's something that I like to do when I'm stressed? Well, something I like to do is snack on some chocolate, maybe snack on some ice cream. And I realized that when I'm in a stressful state, I'll have to eat quite a bit of that highly palatable food in order to really get kind of a stress relief, if you will, to really kind of feel better. And so after I started to think about this, I actually thought it kind of made sense that the mice that had this stress experience in early life are really preferring the sucrose water because they're having to consume so much to get that kind of relief. Now, the reward system kind of studies, this particular study was published in 2018, and I've kind of dropped off working on it, but I'm still interested in the reward system. The disrupted reward system is also indicative of the running distance that is shown on our mice. So I can measure in distance per day, these are kilometers per day, how far a mouse will run in a 24-hour period. And you'll notice here that a little tiny mouse might run 5 to 10 kilometers per day, which is an enormous amount of distance for a little mouse. I mean, I'm pretty proud if I run 5 kilometers or 10 kilometers, and I'm a grown human. But our control mice, they will just run and run and run almost all night long, whereas our stressed mice, they actually are not interested in running. I don't think they get the same kind of endorphin release out of running that the control mice do. And to me, the running pattern disruption and the sucrose preference patterns align with each other. This is a model of kind of the normal stress response, okay, based off of everything I've shown. We have stress, activates the HPA axis. We have higher brain regions and limbic structures that are regulating the HPA axis. There's impact on mast cell activity, neuroimmune signaling with peripheral sensory neurons that communicate up-down with the brain. In a sensitized condition that might be induced by chronic, adult, or early life stress, there becomes this disruption in negative feedback and a more increased tone of positive feedback onto the HPA axis, which can result in kind of a chronic activation, if you will. That can result in a chronic or an over degranulation of mast cells. Neuroimmune signaling increases between immune mediators in the periphery and peripheral sensory neurons. And there is a consistent kind of up tone or a bottom up message of input coming into the brain that we hypothesize leads to a state of central sensitization that may or may not be secondary to a state of peripheral sensitization. Now, if you introduce an intervention such as exercise, or in humans, cognitive behavioral therapy, there is a number of pieces of evidence out there that suggest how this might work. And one of the findings that I looked at was changes in the hippocampus as a result of our exercise intervention. And those findings are consistent with others in the field as well as with humans, and that there is an increase in growth factors in the hippocampus and increase in synaptic complexity, perhaps through increased synaptic signaling within the hippocampus, which might help restore that negative feedback. And then restoration of that can help reduce this state of kind of chronic peripheral and or central sensitization. Which leads me to my current work. My current work now is focused on this intersection of general anesthesia, mood disorder, and reward pathways. Now you might think to yourself, this is kind of a deviation from all the data we just got done showing, but bear with me. By definition, general anesthesia induces unconsciousness, analgesia, and muscle relaxation. And it does so to such a degree that patients cannot be aroused. General anesthesia involves the induction, the maintenance, and then the reversal of that state. But what is the impact of general anesthesia and the stress of that on the brain? And that is really what is quite unknown. So if we have a drug-induced loss of consciousness, even with painful stimulation, the ability, in the ability to maintain, respiratory function is usually impaired. Cardiovascular function can be impaired. General anesthesia is a huge stress on the body, a huge physiological stress, if you will. Not to mention, is the person enduring whatever physical event has occurred that requires the need for them to undergo a major surgery, such as illness or trauma or whatever is going on. So the overall question that I want to ask is this: what is this impact of the general anesthesia and that associated stress on the brain? And in the context that the general mechanism of how anesthesia works remains unknown, leads me to a couple of hypotheses. First, the brain regions that are most commonly studied by kind of the molecular mechanism of general anesthesia is the reticular formation, prefrontal cortex, and the hippocampus. And here's that hippocampus again. The main mechanisms by which it's thought to work is through stimulation of GABAergic and glycinergic activity, which are inhibitory neurotransmitters. So if you stimulate an inhibitory system, you might, quote unquote, shut the brain off. Hyperpolarization of NMDA receptors and inhibition of nicotinic acetylcholine receptors as well. And this mesolimbic system is really where stress, reward, mood, and now general anesthesia all intersect. So this is thought to involve these brain regions in particular. There's also kind of this general belief that it has widespread effects on the entire brain, but this mesolimbic system is key here. And since we have such an overlap in the areas of the brain that regulate the stress response, particularly negative feedback through the hippocampus and the prefrontal cortex, reward, mood, I started investigating and thinking about these lesser recognized effects of general anesthesia on cognitive function. And I found that there's this wide experienced, yet widely under-recognized syndrome called post-operative cognitive dysfunction, or POCD. And while there is some recognition that mood disorder can manifest, particularly depression, following exposure to general anesthesia, its incidence is primarily understudied because the focus of surgery is usually on the recovery of the individual and the reason why they underwent surgery in the first place. So there remains this need to investigate the impact of general anesthesia on mesolimbic and limbic regulation, not only within their function, but also on the stress axis. And so my current work really involves two aims, a preclinical aim and a clinical aim. The preclinical aim is to investigate markers of neurotransmission in the limbic and mesolimbic brain regions that are known to be impacted by depression. And I'm doing so using our collaborators who have animal labs at University of Kansas Medical Center. They have agreed to essentially take control mice, give them a couple hours of general anesthesia, and then a month later test a behavioral test that is indicative of depression-like states in animals. And that test is called the nest building test. So this is a picture up here in the upper right corner from Nature Protocols showing a graphic depiction of the nest building test. So when a mouse is in a normal state, it should take a square cotton nestlet and rip it up into a nice little comfy bed for itself. But a mouse that has all kinds of behavioral changes will not be interested in building that same kind of nesting material for itself. And so we can use this as a correlate of mood disorder in mice. And then we take tissue collection, out of which I'm going to be looking at mRNA expression levels of various different parts of the signaling pathways involved in the stress response regulation, as well as in mood disorder, and in a couple of different brain regions known to be associated with reward, the HPA axis, regulation, and general anesthesia. The second aim is to look at the clinical side of this, and that is to test the hypothesis that, and essentially get an idea of what's the incidence of depression that occurs in patients who otherwise recover as expected from their normal or routine surgical procedures that do need to involve general anesthesia specifically, and not just sedation, but specifically general anesthesia. So this project is looking at enrolling patients who are scheduled for a relatively routine outpatient type of surgery that involves general anesthesia, and then do some outcome measures that evaluate scales of depression and physical recovery. This study it's in progress. We have lab supplies on order. We need to get our brain samples from our collaborators, and targeting next year for our IRB application. And then finally, I'll conclude with our future directions and opportunities for student involvement. So the clinical arm of the, we have an IRB to write. That's a big task. So in terms of involving students, if that is approved, and once it's approved, we have all of these steps to go through. The preclinical arm, and this is where we're at. We essentially need to get our brain tissue from our collaborators and put those through a PCR machine and start looking at gene expression and mRNA expression and see what we can find. I do have some non-related studies ongoing in medical education. I'm not going to go through them, but I just wanted to mention them. There is a study funded by IAMC that I do need some student support for. It is it's concluded and in analysis phase, and these are the steps that need to be taken next. I have a study that is in progress, if you will, and it is currently funded by the Mamie Johnston Creative Art and Teaching Award. And then I have just general ideas that I could use some student help with in just terms of just writing some papers. I have some, whether it's a review or original or op-eds or descriptive paper. I do have just some thoughts, but not enough time to put it to a keyboard. So if you're a student and you are interested in working with me, I will ask you to click on this hyperlink, The slides from this presentation and the recording will be available in the MKRC profile page. But click on this hyperlink, it will take you to kind of a questionnaire that will lead me to understand more about your interests and what you're interested in doing and what type of project is something that you might be willing to work on. The front page of the MKRC Canvas course is shown here on the right. And if you just go and you find my picture and name and you click on it, that's where you'll find access to all of this. And with that, it is 5 o'clock and I am so appreciative that you are all still with me on a Friday afternoon. So thank you all. I will be happy to stick around and take any questions if there are some. Thank you very much.
Thank you so much, Dr. Pierce. Sorry for my brief technical issues at the beginning. Does anyone have any questions?
Yes, actually, I am using my phone, so I apologize. One, that I didn't get to view all the pretty pictures in the presentation that I'm sure that you had, Dr. Pierce. But also, since I'm not in the chat box window, is it gonna be on the MSRM page as well, the hyperlinks for the questionnaire? If not, can we make it so that it is?
That's a good question. I can understand if you said MF or something, but did you mean the MKRC?
Yes.
Okay, yes.
Those letters. I'm dyslexic today, I guess.
Yeah, so if you go to the front page of the MKRC, there are pictures and links to a number of different faculty conducting research in collaboration with the MKRC. You just click on mine, that'll take you to my profile page and I'll have these slides there, okay?
Okay, perfect. Thank you so much for what you do. We've been in email before and just wanted to say publicly that your research is really awesome. So I'm excited to work with you. Hopefully you like me as much as I like you.
You're a phone number, so I can't really tell who's speaking, but
oh, I'm Diana Mentia.
Okay, okay, wonderful. Thank you so much. Dr. Agbas, how are you today?
I'm doing fine, Angela. Thank you very much. Really, it's nice presentation, nice data, nice publications too.
I wish I could go through so much more.
Yeah, you can.
So
absolutely.
much more.
You can. You are young enough. You can do that. I have a kind of naive question. In the early child stress, whatever the initiators, whatever the cause, somehow imprints in the hippocampus where the memory occurs. I'm just curious, later in the life, at animal model, or maybe in a human model, for example, later in the life, and then, for example, in the early stage, maybe the stomach ache or some cramp and happens, and okay, life takes a pull, and everything is pretty much, the leveled up. But the later in the age, if very similar pain inducer happens, like a stress form or something like that, whether this kind of the episodes can be, the so to speak, withdrawn from the hippocampus and then feel the, the same thing. In other words, the childhood in the early, let's say that the family separated, had a cramp and always nervous, and then later in the age, 40, 50 years, the later, if something has happened that very similar, that in his or her early childhood, whether this kind of pain unexpectedly, been exercised. I'm just curious about that. I don't know. Probably I'm just, talk to the pretty, the nebulous, as Dennis says, that the early stage, the child has a trauma, and then that's somehow relevant to the, let's say, stomach ache. Okay, and then these things is imprinted in the hippocampus. Okay, and then this is the early memory, just stays longer, right? And then later in the stage, lifespan, let's say, the 50, 40 years, 30 years, 40 years, if the same stress, that happens, whether the individual exercise the, let's say, stomach pain.
Well, I'm not sure I understand what your question is, sir. But if there is I think you're speaking to the second hit hypothesis, whereby, you have some sort of early life trauma, early life stress experience, kind of life goes on as normal, and then something major happens, a major life event occurs in adulthood, whether it's death of a loved one, a serious illness, or experiencing a trauma, a natural disaster, whatever it is, and then suddenly have a manifestation of a visceral or a chronic pain disorder. Is that all rooted in the hippocampus? Is that essentially your question?
Right. I mean, the hippocampus it's in there, but whether the person exercise or person feels, the very similar pain that what once when he did in the early childhood. For example, parents separated, okay, and then the child had the very chronic, the stomach pain, okay, and then later in the life, and then when the person, to see the very same thing, that it's like the coming back and then feeling the very same thing, and then either the being witnessed, the family separated and little child is just suffering, so he or she just puts himself, in his shoe, and then go back to the early childhood. Once he exercised, he began to feel the pain. It's not that I'm not saying that it's kind of the emotional or the empathy, but it's a kind of real pain.
Yeah, I believe it is. Yeah, definitely.
Okay. So it's a pretty permanently imprinted in the hippocampus, so we can't really erase it, because the early memories stay longer. I know that.
Yeah, exactly.
Okay.
Dr. Wolf.
Yeah, hi. I really enjoyed this a lot. I guess I'm looking up at my screen here. Sorry about that. But I had a couple of comments. So one thing that was interesting to me had to do with the running wheel and stuff like that, and your sucrose. And so Edwin Gluck, I don't know how he pronounces his last name. He's my advisee, but he was telling me how they had to stress rats in a study that he did in undergrad, and stressed rats preferred glucose. And so you might learn something by talking to him, but I had friends that had to stress rats in order to get them to drink alcohol. So normally they don't like it, but if you stress them, then they'll go out and they'll be more attracted to alcohol or cigarette smoke. So that was one thing that I just thought was interesting. But I had a couple of questions. And so number one that was kind of fascinating to me related to the degranulation of the mast cells. And if you know whether or not you need somatic or autonomic nervous system innervation for that to happen, or what you think is actually triggering that mast cell degranulation.
Okay, that's a great question. There's probably, I haven't kept up with it too well, admittedly, but there's probably denervated studies out there that look at what is dependent on that mast cell degranulation state. The work that I am most familiar with indicates that it is antidromic signaling from peripheral sensory neurons in conjunction with the presence of glucocorticoids, because the mast cells themselves do express to the corticotropin releasing factor receptor, and they express glucocorticoid receptors, both of which have been shown to induce degranulation upon exposure. The kind of persistent chronic states, because they will regranulate and kind of, stabilize over time, but that kind of chronic state has been shown to require that peripheral sensory innervation. So if you denervate using, say, a peripheral suture, or you ligate, say, some sort of sensory nerve, then that can disrupt that neuroimmune signaling, but it is antidromic effects.
Okay. The other thing that I thought of here had to do with your exercise wheel and things along those lines. And for those of us that work with animals, you always know that when you get a new batch of animals, you have to let them acclimate.
Oh yeah.
And oh, they must acclimate. And it seems to me that that whole acclimation response, whatever that may be, could be a part of this whole thing here. And it's always intrigued me what is actually responsible for that. The other
kind of the novelty effect, right? I mean, you introduce
Yeah.
something new and they're really interested in it because it's novel. Yeah, I definitely was always very careful with acclimatization protocols and measuring what our findings based off of the proper in the field accepted acclimatization kind of timelines, definitely. Yeah.
Well, we actually, I was involved in a study where we were bringing in, it was a summer NIH sponsored summer study, and the rats didn't get there when they were supposed to. And so we had to study them acutely. And so my colleague, long history of experimenting with rats. And so we had our drug dose response curve stuff all set up. And they were more than 100,000-fold more sensitive to our our norepinephrine, endothelin, whatever else, I can't remember what we were using, but they were just amazingly sensitive that the lowest doses were given the maximum response already. So there is very definitely something there. Along those lines, though, also is I was not necessarily, I'm not in that field, so I wasn't familiar with the urocortin receptors, but it looks to me like they're G protein coupled receptors. And so the things that I've been interested in with my colleague Yaping too, is this notion that we measure receptor levels and we measure hormone levels, but there's the regulator of G protein signaling proteins, all these things that actually impact in the cell to determine whether or not you're actually the magnitude and the duration of your response. And so I was curious if you had functional readouts, just looking at mRNA. In our hands, many times there was a wide variation in the protein and the response relative to the mRNA that we had measured.
That's a fabulous question. And spoken like a true pharmacologist. I kind of get to the cell membrane and then stop. So my anything small outside of the cell membrane, I kind of like, once you get inside the cell, I kind of get a little, I'm not quite so interested in it. But yeah, I definitely I have to point that out as a caveat, is these things might be changing their mRNA expression pattern, but that doesn't necessarily mean that it's the causation of these things. It's just a snapshot of one step in this process. So in terms of looking at the different, cofactors or co-signaling mechanism, co-signaling, the downstream pathways, I never got into that kind of thing. I would say that I did start to kind of get into calcium signaling and, upon exposure of different ligands, but it just, the behavioral field, they were like, yeah, okay, yeah, they want to know more about the interventions and what works and whatnot. But if I had the time to go into those kind of intracellular signaling pathways and look at all the different components and start piecing them together, I definitely would. Yeah, I just didn't have the time. But maybe that's something I could do now. I mean, I have, obviously at KCU, we don't have animal vivariums. So I've been kind of brainstorming for the next iteration of my intramural grant. What can I do with cell lines? And I've never really used cell lines before. The only thing I've ever done with cells was I plucked out peripheral sensory neurons from dorsal root ganglion cells and would plate them and do different stuff. But I mean, they only lasted like a couple of days and for very short-term studies. But in terms of like ordering cells and doing cell line stuff and then just start squirting on ligands, I've never really done any of that, because if I wanted cells, I just took them out of mice, right? So I have been thinking about what to do next. And so I might have to hit you up for ideas on that kind of thing, because that's definitely new to me.
Well, I think you and I are both in the same field there. I mean, that's me too. I don't study cells. I study animals. Yeah. I study organs.
Yeah, exactly. All right. Well, thank you so much. And thank you, Anne-Marie.
Awesome. Yeah,
great.
thank you so much again.
Enjoyed it. Bye.